Let's start off with mitosis. This little piggy represents a cell, and the saddle on its back represents the copied DNA that it carries in its nucleus. The first phase is the prophase. The orange blocks are the nucleus, which are now disappearing, and the blue blocks are the chromosomes. The magenta blocks is the cell membrane. The centroles gather at the poles of the cell to form spindle fibers across the cell. The chromosomes are still floating around. In metaphase, the spindle fibers organize the chromosomes in the middle of the cell. In anaphase, the spindle fibers pull apart the chromosomes so they are no longer together. In telophase, two new nuclei are formed around the two groups of chromosomes. This is the beginning of two new cells. We now visit Mr. Cytokinesis to split our two new cells. In the end of mitosis, we are left with two body cells that have no extra DNA on them. Let's fly over to meiosis. This little piggy still has a saddle, so we know that it carries copied DNA to start off meiosis. In prophase 1, we still have our disappearing nucleuses and our chromosomes, but our chromosomes are now numbered in 2 and 3. These are not the only chromosomes we have, but they are the ones that I'm going to be using as examples to explain this process. And the fences in this situation are the cell membrane. The centroles still gather at the poles, and the spindle fibers do the same thing they did last time. The chromosomes 2 and 2 find each other because they like to be with homologous chromosomes. Chromosomes 3 and 3 also find each other. They have the same number, but they are not the same. That's why they're different colors. Here we see that the sheep are now different colors, which is a mixture of their previous colors. This is what is called crossing over in meiosis. So these sheep are now green, and these sheep are now pink. Then we move on to metaphase 1. These sheep are now pushed to the middle because of the spindle fibers. Now we move on to anaphase 1. The sheep are now separated into two groups. As you can see, the sheep with the same number chromosomes cannot be in the same group. We move on to telophase 1. The chromosomes are still in their groups. The only thing that is different is the nucleus forming around them. We end up with two daughter cells, just like we did last time. But we do this process again, but with a normal cell, which has no extra DNA. This is prophase 2. The nucleus still disappearing, and we have our now normal chromosomes with no number. And the fences are still our cell membrane. In metaphase 2, the centroles do their same job, so do the spindle fibers. The chromosomes are pushed in the middle. In anaphase 2, you can see that the chromosomes are now chickens, because in each group there are only 23 chromosomes. The groups are now smaller because of that. In telophase 2, the nucleus form around the chromosomes. Lastly, we end off with 4 gametes, which are sexual reproduction cells that can be found in the eggs of a woman and the sperm of a man.